study them. Besides the tables that I've told you, you do not need to memorize the content of those tables. And besides, I won't give case-based questions. Okay, case-based questions you will not have in your exam. The other thing, now, after those lectures you've had these days, this week, now the immunology is more difficult for you or more easy? Does anything changed for you after these lectures?
We will have a basic case.
No. I don't care any case-based questions. Yes. Just. So you will have multiple choice questions. So after the lectures, tell me, is the immunology now after the lectures of this week, is the immunology easier for you or more difficult for you? Or what? Did you understand? I've given you my number, my phone number. You can ask me scientific, anything related to the topics on WhatsApp. But please do not ask about the exam, about the scores, about the points, anything. Just send me your assignments by your name, just 3 lines, not more. And ask any question if you have about the slides, about the explanations, about anything. I will explain you by voice or anything that is possible, by figure, if you have questions. Okay, the last lecture. Immunodeficiencies. So again, I'm not going to talk about the different types of immunodeficient diseases. I'm just going to talk about the basic immunology of some immunodeficiencies. So the course is basic immunology. You will have again some lectures about the diseases that today, both in hypersensitivity and for immunodeficiencies, I have talked to you in summary. You will have a complete lectures about them in your clinical courses. So, what is immunodeficiency? What does that mean? What does immunodeficiency mean?
We don't have appropriate immune response.
We won't have appropriate immune response or proper immune response. Why?
Because we have not sufficient immune response.
The reason, yes, is that we do not have sufficient or normal immune response. Why we do not have?
Maybe because we have deficiency in T cell or B cell or both of them.
Yes, we might have deficiency in T cell, B cell, or both of them. But why we might have deficiency in T cells?
Maybe because genetics or
Because of genetics, genetic mutations.
is
Most mutations, we might have mutations in the genes that are involved in T cell production, B cell production, T cell activation, B cell activation, in cytokines, in receptors, in complement proteins. So many genes are involved in immune responses. And any mutation in those genes can cause immunodeficiency. They are called congenital immunodeficiencies. And we have acquired immunodeficiency as well. Like AIDS or HIV or some other conditions that they are not, those immunodeficiencies do not have genetic base. They have some acquired, some other reasons that are not genetically inherited for us, like HIV or some other conditions. So we are going to talk about congenital or inherited or genetic mutations related immunodeficiencies, and a little about acquired immune deficiencies. So, it is important to detect the type of, so what would be, let me ask the question another way. Now, so you know that the definition of immunodeficiency, having no normal immune response, having insufficient immune response, okay, it's immune deficiency. What would be the consequences of immune deficiency?
Infection.
Infection. What else? So that's logical. If we do not have normal immune response, we will have higher chance of infectious disease because the function of immune system is to prevent infection.
Autoimmune.
What else?
Autoimmune.
Why autoimmunity is possible?
We don't have an immune response to prevent the infection being this disease.
Because infectious agents are the important cause of autoimmunity. And sometimes, do you remember that we have a part of immune system is regulators of immune system. Immune regulators including regulatory T cells, complement regulators, macrophage B, M2, regulatory cytokines. They are suppressory arm of immune system. So when we have deficiency in those components, we might have autoimmunity. So immune deficiency can lead to infectious disease, autoimmunity, what else? Tumor. One important function of immune system is to fight or to prevent cancer. So when their immune system cannot work normally, so we might have higher chance of tumor or cancer. So, the third question. In all types of immune deficiencies, we might have all of these diseases. I mean, if we have any kind of immune deficiency, the consequences would be autoimmunity, infection, and tumor. And you know that we have multiple types of infectious diseases. In all immune deficiency, we might have increased risk of fungi infection, intracellular bacteria, virus, or not.
Depend on the deficiency in where, which cell.
Okay, so based on the type of the deficiency in the immune cells, we might have different consequences. For example, you know that the main immune response against viruses is, what is the main immune response against virus? Cytotoxic T cells. So any deficiency in cytotoxic T cell will increase the infection with viruses. So the main immune response against extracellular bacteria is, and what other cells? Macrophages, phagocytic cells and complements. So any deficiencies in complements and macrophages will lead to increased risk of infectious with extracellular infection disease. Which part of the immune system is responsible for cancer fighting or cancer prevention? Cytotoxic T cell is the most important one. So when we do not have enough CD8 T cells, the risk of cancer will, you know all of this. You should just think about the things you know. Intracellular, extracellular, humoral. So the main immune response against, the main adaptive immune response against extracellular bacteria, adaptive immune response against, not innate. Antibody, humoral immune response. Humoral immune response and production of antibody is the most effective arm of immune response against extracellular bacteria and fungi. So deficiencies in antibody production, so any deficiency related to B cell, both B cell production and both B cell activation can lead to increased infection, increased risk of infection with extracellular bacteria and fungi again.
I have questions. What's the role of this T17
T helper 17. T helper 17 is very important in extracellular bacteria and fungi and mucosal surfaces immune response. If we have deficiency in T helper 17, we might have increased risk of infection in mucosal surfaces and immune response against bacteria and fungi. Is that clear? So, if you know that in immune system, we have innate immunity and humoral and cellular immunity of adaptive immunity. That's the whole component of immune system. And you know the infectious agents and tumor nature and the type of immune response against them. So if based on the type of deficiency, we might have increased risk of different infectious diseases or different diseases. For example, if the defect is related to humoral or B cell, we might have pyogenic. What does pyogenic mean? Fever. The infectious agent that can cause fever. Or a pus, they can form pus. Pus means a fluid, a thick fluid containing dead cells, dead leukocytes. So pyogenic and encapsulated bacteria. For example, Streptococcus pneumonia is an encapsulated bacteria. And some viruses. Isn't it weird? We have deficiency in B cell, but deficiency in B cell can increase infection with some viruses. What's the reason behind it?
When it turns.
Yes, one important function of, thank you, got it. One important function of B cell is the main function of B cell is production of antibody. And one function of antibody is neutralization of the viruses. So when there is no neutralizing antibody against viruses, so in B cell deficiency, the infection with some viruses will increase. T cell defects, we will have, of course, viral infections increase, we will have increased fungi infections, intracellular microbe and fungi intracellular, and viruses. What does?
If we have B cell deficiency, it will affect our B cells?
Very good question.
Of course.
Very good question. If we do not have proper T cell defect, doesn't it affect humoral immune response? Of course it affects. When there is no normal T cell response, we won't have class switching. I'll talk about it. We won't have class switching, we won't have memory formation, we won't have affinity maturation. Yes, that's correct, thank you. I'll talk about it. What does this mean? Combined if defects,
T cell and B cell.
combined having defects in both of them, both arm of adaptive immunity. So this one would have, of course, more severe form of the immune deficiencies. So we have broad susceptibility to bacterial, viral, and fungal pathogens. And we might have innate defects. When we have innate defects,
Defect in macrophage.
macrophages and complement are the most important. When we have defect in macrophage, so one of the important function of macrophage is phagocytosis of extracellular bacteria and fungi. So we will have some problem with extracellular bacteria and fungi. And of course, in innate immunity, what else we have except than macrophages? What else we have in innate immunity? Complement system. And deficiencies in complement system have its own consequences. That's what I talked about. So based on the type of the component of immune system, we will have different types of manifestations or higher risk of different infectious diseases. And some other clinical clues that they are more general, including increased risk of opportunistic infections. What does this mean?
Bacteria that in normal individual people can cause any infections.
Diseases, yes.
That's like people have AIDS, maybe can have.
Yes, thank you. So opportunistic infections are the infectious agents, microorganisms that in normal situation, they are not pathogenic, they do not cause disease. But when we have immune deficiency as a result of HIV or other reasons, those opportunistic infections have a chance, opportunity to cause disease. So the opportunity is indeed depends on the immune system status of the people. So it's very important. One of the main symptoms of most of the immune deficiencies is recurrent severe infections. Repeated severe infections over time is a common symptom of most of the immune deficiencies. That's normal. When we do not have immune system, recurrent infections would be common. And I talked about autoimmunity, and of course malignancies or cancers, especially virus-associated tumors. Do you remember I told you that viral infections are among the important inducer of cancer formation or transformation. So when immune system cannot kill the viruses, so those viruses have chance to induce malignancies or cancer. So the type of infection, it's important, the type of infection is the first clue to detect which arm of immunity is defective. The type of infection, I talked about it. So this is the comparison between B cells and T cells immunodeficiencies. For example, in B cell deficiency, we won't have humoral immune response and antibody detection, but in T cell deficiency, we have cell-mediated and, of course, antibody-dependent immune response defection. You know the reason behind it. And the type of infection, pyogenic and encapsulated bacteria, but for T cell, we have viral and intracellular infection, higher risk for that kind of infection. Serum immunoglobulin. What is this? Serum antibody is checked in medical laboratory. So in B cell deficiencies, we have reduced serum immunoglobulin. But for this T cell is different. In some T cell deficiencies, we have normal antibody. In some others, we have reduced, or the type of antibody will change. And lymphoid tissues in B cell and T cell deficiency, for example, we have some problem in lymphoid tissues, and of course the clinical consequences would be different. So immune deficiencies are categorized in two main groups. Primary immune deficiency or secondary immune deficiency. What's the difference between them?
Primary acquired.
Primary, no.
Primary genetics. Congenital.
Yes, congenital. Primary is indeed congenital or inherited disease, genetic based, and the secondary is acquired or something that is not genetic or congenital. So let me skip all of them. So there are different defects in innate immunity. Chronic granulomatous disease, I'll talk about them, some of them, almost all of them, one by one in the next slide. This slide is a summary of what I'm going to talk about in the next slide. For example, in this disease, chronic granulomatous disease, we have a problem in ROS production. What is ROS? What is ROS?
Oxygen.
Reactive oxygen species. What is the role in immune system?
The way that macrophages and other phagocytes.
Phagocytic cells, yes. Kill the bacteria. So ROS is reactive oxygen species that they are produced by macrophages and neutrophils and other phagocytic cells, and those cells use them to kill their phagocytosed microbe. Or we have some deficiency in leukocyte adhesion and migration. I'll talk about them. We have some problem in lysosomal trafficking. What does that mean? We have some deficiencies in lysosome trafficking. What will happen if we have a problem in lysosome trafficking? What is the role of the function of lysosome in immune system?
Bind to the phagocytes.
Yes, not phagocyte. Yes. When a phagocytic cell, a phagocytose, a bacteria, for example, those vesicle is called phagosome, and you know that this phagosome is merged with lysosome. And inside the lysosome, there are so many enzymes and toxic agents that they kill the bacteria. So again, we will have a problem in phagocytosis here. And NK cell deficiency is clear, and let me move the others. So the first disease is chronic granulomatous disease. The main problem, this is, you know that this is a macrophage, this is phagosome and the pathogen inside it, and this is lysosome, and binding of lysosome with the phagosome. We have phagolysosome, and they are the toxic agent of lysosome, that they are toxic for the bacteria. And they are, this was oxygen independent killing of the bacteria. But there is another mechanism which is related to oxygen or ROS dependent killing of the bacteria. In chronic granulomatous disease, patients have problem in the production of ROS. Okay, so when these cells, phagocytic cells, cannot produce ROS, the bacteria the killing capacity of those cells will decrease, and the bacteria will get alive inside the cells. So you do not need again to memorize the details of the deficiencies. For example, this NADPH oxidase is involved here to produce ROS, but it's not needed to memorize them. But knowing that we have problem in ROS, and if you know what would be the consequences of ROS deficiency is enough. So we have ingestion of the microbes, but the phagocyte cannot effectively kill them. So I told you in DTH, whenever macrophage or phagocytic cells cannot kill the ingested microbe, they will make granuloma. Did you remember? Granuloma was made by macrophages. That macrophage, after the ingestion of the microbe, cannot kill it efficiently. So it makes granuloma to restrict the spread of the bacteria to the other tissues. So here we have granuloma again. It's called chronic because we have a genetic deficiency, granulomatous disease. So we have higher risk of pyrogenic bacteria and extracellular bacteria and fungi infection. You do not need to memorize those and this one. So most of the congenital immune deficiencies begin from childhood and sometimes from infancy. So it begins like other congenital diseases in childhood, and we will have recurrent bacterial and fungal, mainly extracellular infections. For example, Aspergillus is a major invasive pathogen for this disease. That's it. So we will have granuloma formation here. It's important. You can see. Can you see here? It's a granuloma, a mass of immune cells. This is a mass of macrophages and T helper maybe cells, and you can see here again different masses with different size. That's why it is called granulomatous infections. So it's a very chronic granulomatous infection in the nose of the patient. So you can see here granuloma, a mass of macrophages around the bacteria or any foreign substances that macrophages cannot eliminate it efficiently. Another disease is called leukocyte adhesion deficiency. What does that mean? Adhesion deficiency. We have adhesion deficiency.
The leukocyte, when the infection occurs, the leukocyte must came from the long distance and must adhere and stop at the infection site by the some like signals from the infected cell or by the adult.
Thank you. Thank you. You do not need to again memorize the details of the difference. We have different types of leukocyte adhesion deficiency. I have a figure. I'll talk about it in the next slide. But first of all, you do not need to memorize the differences between the three types of LAD. Just knowing that we have adhesion deficiency is enough. But those clinical clues are important. For example, those patients that indeed, let me skip for the next slide and then I'll back to these slides. Here, they are the endothelial cells of the blood, and they are the lymphocytes in the blood circulation. If you remember from inflammation, this is the blood vessels and leukocyte inside in the blood circulation, and this is, for example, a skin, and again an infection with a bacteria. So we need a migration of leukocytes from blood vessels to the site of bacteria. This migration is mediated by some adhesion molecules around here. The leukocytes, for example, if I am a leukocyte and if I want to get out of this door, I need to catch the door, open the door and get out of the room. This is my adhesion molecule. So in this disease, cells have problem with their adhesion to the blood vessels and their movements. So it is important to know that we have neutrophils. It's very important note. We have neutrophils in the blood, but they cannot efficiently reach the infection site. Neutrophils and other leukocytes, we have them in the blood. Their production is normal, but they cannot efficiently reach the infection site. So we won't have effective immune response. As a result, we will have recurrent again bacteria and fungal infection. We will have leukocytosis. What does it mean? Leukocytosis. Increased number of leukocytes in the blood. Increased number of white pieces in the blood. And the patient will have wound healing, problem with impaired or problem with wound healing. It's important to know that one important function of mainly innate immune response is wound healing and tissue repair. When a patient cannot have proper immune response, cannot proper cells movement, so the wound healing won't be done properly. So the rest of the, for example, rolling, no, that's enough. So this clinical manifestation is enough to know. Question, comment? Another disease is Chidiac-Higashi syndrome. Chidiac-Higashi is another immune deficiency that in this patient we have problem with lysosome trafficking. or vesicle trafficking. And you know that those patients, most of them have albinism. Let me find here, this one. Because you know that the pigments that are produced by the cells of the body, they have to secrete it to the outer membrane, out of the cells. When we do not have normal vesicle movement, so the patient cannot produce any color. So they will have albinism, and they will have defective phagolysosome formation. And again, we will have defective immune response against extracellular bacteria and fungi in neutrophils and macrophages. And we have leukocytes with the granules are larger than normal inside the neutrophils. It's a neutrophil, but the granules are too large. That's why it is said we have giant lysosomes inside the leukocytes. Giant means very big, and we have them here. And of course, we will have problem with NK and CTL cytotoxicity. Why? In Chidiakigashi, we have lysosomal or vesicle trafficking problem. All vesicles, not just lysosomes. So those kind of defects can affect NK cell and CTL activity. What's the reason behind it?
Because it's effective presentation.
No.
Because we must make processing to the protein to make peptide.
NK cell do not need processing.
I'm talking about the CT.
Yes, might be, but dendritic cell might, yes. One reason could be that. There is another reason. One reason could be that for CTL that there is no proper antigen presentation. What other reason? Ladies here, please. Why lysosomal or vesicle trafficking?
What mean trafficking?
Movement of vesicle from inside the cell toward outside the cell. To release the content of granules out of the cells. In mast cells, we have it. In eosinophils, neutrophils, there are granules. Okay? So, why, when we have vesicle trafficking problem, we will have NK cell in proper immune response? Because inside the NK cell and CTL, we have granule as well. All the cells that have granule, we have inside them granule. Do you remember? And inside the granules of them, there are perforin and granzyme. So when those process doesn't work normally, they cannot release the content of their granule on the target cell. And of course, it affects nervous system as well. It doesn't matter. You do not need to memorize this. And some patients have bleeding disorders, uncontrolled bleeding, because their platelet cannot work anymore. So when we have a genetic defect that affects vesicle trafficking, any biological activity in the body that is dependent to vesicle trafficking would be defective, including NK cell activity, CTL activity, macrophage activity, neutrophil activity, all granulocytes activity. So, complement deficiencies. For example, if we have deficiency in C2 or C3, I've talked about you, that if we have deficiency in a classical pathway complement, we will have higher risk for systemic lupus erythematosus. What's the reason behind it? If a patient has a deficiency in classical pathway complement,
What is this?
SLE, I have talked about it several times. It's lupus. It's an autoimmune disease. Immune complex related autoimmune disease. Systemic lupus erythematosus. Shortly it is called lupus or SLE. Why, when a patient, the question is this, it's an exam question. It's an exam question. I have focused on this matter several times. When I talked about complement, I have talked about this. When I talked about hypersensitivity, I have talked about this. When I have talked about autoimmune disease, I have talked about, and this is the fourth time that I'm talking about SLE and the immune response responsible for that. Why? When the patient has deficiency in complement, it's the risk of SLE will increase. The question is this. If a patient, the question is this. Why? If a patient has deficiency in the complement proteins, classical pathway of the protein, of the complement. A patient has deficiency in those complement proteins. The risk of SLE will increase in the patient. What's the reason behind it?
Because of the B cell, because complement system activates B cell on
Think about immune complexes. No.
Because deficiency of these proteins will not activate complement system, and macrophage will not bind with the LC receptors, and will not clearance of antibody.
Clearance of immune complexes. Thank you. We won't have one important function of when we say, have a look here, Haji, have a look here, Haji, have a look here. When we have deficiency in complement, you do not need to rely on your memory. You should think about the function of complement. If you think of the function of complement, you will find the problem. One function of complement is clearance of immune complexes. When we do not have normal amount of complementary proteins, so the clearance of immune complexes will not be done properly. And when we have a higher immune complex, it can cause SLE. It's an SLE related disease. Immune complex mediated disease. Here, it's the fourth time I'm talking about these diseases. What can I do? Khudaya, you killed me. And deficiency in C3, they are the complement members of classical pathway. And deficiency in C3 is related to pyogenic bacterial infection. It's logical, you should know that. And sometimes, let me skip this one. And so again, we might have some problems, deficiency in classical pathway, so autoimmune disease risk will increase. If we have alternative pathway, pyogenic infection will increase. And if we have late pathway, we will have problem of both of them. There is another deficiency. I've talked about hereditary angioedema or hereditary angionucleotic edema. It's related to C1 inhibitor, a regulator of complement. When there is no regulator of complement, we will have unwanted complement activation and unwanted inflammation, and it leads to destruction of the vessels and edema. And you can see the edema here, angioedema. So, there is a disease which is called formation of, I do not know why the name of the disease is missed from here. It leads to the formation of a disease which is called PNH, proximal nocturnal hemoglobinuria. Those are urine. This is a normal urine. This is a urine that why it is called hemoglobin urea. What does that mean? Hemoglobin urea. Having hemoglobin inside the urine. Hemoglobin. When we have hemoglobin in the urine, it is called hemoglobin urea. Here we have hemoglobin in the urine as a result of uncontrolled complement activation on the RBCs. So we have uncontrolled lysis of the RBCs, and when we have uncontrolled lysis of RBCs, we have increased release of hemoglobin, and hemoglobin is would be question. We will have hemoglobin in the urine. Is that clear? It is called proximal nocturnal hemoglobin urea. It's a problem with another complement regulators. So we have intravascular hemolysis, RBC lysis in the vessels. Now, adaptive immune deficiencies. Until now, I have talked about some examples of innate immune deficiencies. Let me check the time. Okay. Get back in 10 minutes. Get back in 10 minutes. You should remind me. You should remind me that it's the rest time. Get back. Different defects in phagocytic cells we talked about, including lysosomal defections, ROS production defections, or leukocytes movement, migration deficiencies, adhesion deficiencies, and complements. Now, I will continue the class with the deficiency in adaptive immunity. The deficiencies in adaptive immunity could be categorized in two groups. The first group is about the deficiencies that affect development of the lymphocytes. Deficiencies that affect development of the lymphocytes. And then the second group are the deficiencies that affect the activation of the lymphocytes, both T cells or B cells. So, in the development of T cells and B cells, the deficiency may be for T cells and B cells together or some of them. One of them indeed. You have a look here, you've seen this in the production of T cells. We have a hematopoietic stem cell, and from one hematopoietic stem cells, we have T cell development and B cell development in different sequential steps until the mature T cells or B cells. And many genes and proteins are involved in the development of T cell and B cell. Okay, many genes are involved in the development of T cells and B cells. Some of them are similar for both of them. I've talked about some of them. Can you tell me an example of enzymes that are involved in both T cells and B cells? RAC. TDT, thank you. They are involved in recombination of receptors of T cells and B cells. Both RAC deficiency and TDT deficiency, they are examples that involve both T cells and B cells, and those enzymes were required for VDJ recombination. VDJ recombination is the process of production of TCRs and BCRs that we talked about, the combinatorial diversity, junctional diversity, and production of TCRs and BCRs. So many enzymes are involved in VDJ recombination. So any deficiency in those enzymes can affect both T cells and B cells. Another example is the enzymes that are involved in nucleotide production. For T cell proliferation, we need nucleotide production. You know that for replication for proliferation, we need DNA replication. Do you remember anything about DNA replication? What is DNA replication? information, another copy of DNA for division of the cell into two daughter cells. When we want to, when a cell is going to divide into two daughter cells, it needs to make two copies of all of its DNA. So making of copies from the DNA of the cell is said DNA replication. For DNA replication or making of copies from the DNA, there is need for nucleotides. Cytosine, thymidine, do you remember from biology? So if there is any deficiency related to DNA replication, so we will have a deficiency in cell proliferation of both T cells and B cells again. Or some cytokine signalings, again, I told you that some cytokines are important in hematopoiesis or T cell and B cell development. Interleukin 7 is a good example of the cytokines that are needed for T cell development. And MHC expression is needed for T cell development. Why we need MHC expression for T cell development?
During the development of T cell in the thymus, to prevent autoimmune disease, so we need MHC.
Yes. How? Yes, that's correct.
By ABC.
They are expressed by APC for
Yes.
selection of positive selection
Yes.
and negative selection that prevents the autoimmune disease. We need MHC expression. So without MHC expression, we won't have normal T cell. And when there is no normal T cell, the B cell immune response will be affected again. And let me skip. So when we have a deficiency in both T cells and B cells, it is called combined immune deficiency. And those deficiencies are mostly they are severe. So they are called severe combined immune deficiency. So severe combined immune deficiency are a group of diseases, different kind of diseases that in all of them we have both deficiency in T cells and B cells. Or we have deficiency in the T cell, that deficiency in T cell has affected B cell immune response as well. Okay? And we have severe diseases, in short they are called SCID. So the SCID diseases affect both humoral and cell-mediated immune response. And both T cells and B cells and sometimes NK cells even are affected. Of course, patient will develop life-threatening, lethal infectious diseases with different kind of microorganisms, including bacteria, viruses, fungi, and CMV infection, pneumocystis infection, and pneumocystis juvenile, and persistent diarrhea is a typical manifestation of the most of the SCID immune deficiencies. And it is important that live attenuated vaccines can cause disease in those group of patients. Live attenuated vaccines, what is this? Live attenuated vaccines. There are a type of vaccines that, for example, for polio, we have the virus of polio in the vaccine. The virus is not killed, is live but attenuated. Attenuated means weakened. Very weak. We have a very weak virus in the vaccine that this virus can proliferate, but it cannot cause disease in normal patients. All of us have received polio vaccine, live attenuated polio vaccine. The polio vaccine is a weakened viruses of the polio that can proliferate, but normally it cannot induce disease. But in SCID diseases, those live attenuated vaccines can cause disease, and that's why injection of those type of vaccines is forbidden for SCID patients. And it's very important, and it's logical. Without intervention, it means without treatment, all SCID patients, all diseases of the SCIDs could be fatal for them, even in the infancy, could be fatal in infancy. They need intervention in infancy because after birth they have deficiencies in multiple arms of immune system, and they will have severe infection with many types of microorganisms, including bacteria, viruses, and fungi. So these severe infections could be lethal for the infant if we do not have proper intervention. So here you can see examples again. You are not going to memorize most of them. So RAC1 and RAC2 and some other enzymes and proteins that are involved in the combination of the TCR and BCR, they are examples, and they are involved in nucleotide metabolism, cytokine signaling. This one is called interleukin 2 receptor. I talked about the common gamma chain. There is a receptor for several cytokines, a shared receptor for several cytokines. When a person doesn't have this common gamma chain, it cannot respond to several cytokines. That's why I talked about it. And I talked about MHC expression and thymic development. That's why I'll talk about some of them in the next slide. No question? This topic, some part of this topic is just memorizing, but some others you can find the response rationally based on your knowledge about immune responses and the functions of different arm of immune responses. So X-linked SCID is a very common type of SCID, which is caused by interleukin receptor or common gamma chain deficiency or mutation in common gamma chain. It is called common because it's needed for the function of several cytokines, including interleukin 2, 5, 7, 15, and some others. So when a person have mutation in this chain, it won't respond to those cytokines. So several cytokines cannot work properly. So the patient will have X-linked. Why does it call X-linked?
Because it's the defect on X
The
chromosome.
gene is located on the X chromosome, not other chromosomes. So any mutations that in the genes that are located on X chromosome are called X-linked mutations or X-linked diseases, and this is X-linked SQ. And so when several cytokines are involved, T cells are decreased. This is the sign of decrease. NK cell is decreased, but the B cell number is normal. B cell number is different from function. You should discriminate between them. When interleukin 7 signaling is defective, we have defective T cell development, that's normal, and interleukin 15 is needed for NK cell. And humoral defect, largely we have a humoral defect. Please take care. We have a humoral defect, but B cell numbers is normal. Why? Why the number of B cell is normal, the patient has a humoral defect.
B cell cannot produce antibody, but there's a number of B cell in our body, but they cannot produce antibody.
Why?
Differentiate.
Why? Yes, we have the normal number, but they cannot produce proper amount of antibody because there is no T cell here. The deficiency is on T cells. When we have deficiency in T cells, the B cell number is normal, but the immunoglobulin or the humoral immune response is defective. Clear or not? Because the defect is not on B cell, it's on the T cells. Any defect in the T cells cannot affect the number of B cells. It usually affects the humoral immune response, I told you, they affect the production of antibody, memory cell formation, affinity maturation, class switching, and this is a vaccinia necrosome, again, a gram-pneumococcal infection in a 3-year-old baby with XLA. And those are another examples. You do not need to memorize those. So, the VDJ recombination defects, RAG1 and RAG2 mutations and other mutations involved in VDJ recombination, and you don't need to memorize those as well. So, we have deficiency or absence, severe, profound absence in both T cells and B cells. Because for development of both of them, we need recombination. When the recombination doesn't work normally, we won't have both of them. And there is a syndrome which is called Omen syndrome. This syndrome is a reduced T cell generation, but the eosinophil and high level of IgE will produce the patient. Because there is no T cell, and as a result of absence in T cell, the compensatory system will increase the number of eosinophil and IgE, and this is they have a high level of IgE. That's enough. So, MSC class 2 deficiency. You guess the consequences. What problem will the patient with, will have the patient with MSC class 2 deficiency? You should, first of all, you should think about the function of MSC class 2.
Bind to this T helper cell.
So they are needed for the activation of T helper, all T helpers, not T helper 2.
Yes.
All T helpers. So we won't have normal T helper function. So can you tell me the normal T helper functions, including, let me name the number of T helpers.
1, 2,
T helper 1,
2,
2,
17. Actually regulated.
So any functions related to this, so all T helpers would be defective. So does humoral immunity have defects here?
Yes.
Yes, because we do not have normal T helpers. Does immune response against extracellular bacteria is defective?
Of course.
Is immune response against viral infections is affected?
Maybe, yes, because
Because T helper provides help for cytotoxic T cell and APC antigen presentation. Does immune response in mucosal surfaces is affected? Yes. Is immune response in humoral surface affected or not? Yes or no? Is immune response in mucosal surfaces affected or not?
yes, of course.
Yes, because helper 17 is important, and even the regulatory T cells produce TGF beta, and this one is important for IgA production.
The regulatory also has to be presented by the
Yes,
APCs.
yes. They are T cells. They are CD4 positive T cells. They are CD4 positive T cells. Yes, they need antigen presentation and activation. So, again, whenever you are going to answer, you do not rely on your memory. Think about what you know. If you know the function of the HLA class 2, you can answer many questions about this deficiency. So, it is called Bayer lymphocyte syndrome. First of all, MHC class 2 is needed for T helper 2 development in the T helper development in the thymus. Not only the function, we won't have T helper normal T helper development in the thymus. It's needed for T helper development. So the number of T cells will decrease, T helpers. So we have reduced CD4 positive T cell. both development and function, both the numbers and functions are affected. And of course, T-cell dependent antibody production is affected. You are not going to memorize the type of the mutations. This is for most SCID diseases. For most of the SCID diseases, this therapy is done usually. This is hematopoietic stem cell transplantation. Hematopoietic stem cell transplantation. It's a kind of bone marrow transplantation. We have a patient that its bone marrow, the hematopoietic stem cells cannot produce normal T cells. We will give the patient a normal bone marrow. Clear? Clear? The patient has defective bone marrow or defective hematopoietic stem cell. We will give the patient new hematopoietic stem cell from a normal patient. It is called hematopoietic stem cell transplantation. For most SCID diseases, it's usually done. Another syndrome is DiGeorge. Have you heard about it? DiGeorge syndrome. Patients with DiGeorge syndrome do not have normal thymus. They have thymic hypoplasia. They do not have normal thymus. So again, the consequence is, you tell me the consequences. When a patient has DiGeorge syndrome or a patient do not have normal thymus, what consequences will have for the patient? When the patient doesn't have normal thymus.
We have problem in T cell.
So T cell developments are affected. Which type of T cells?
Yeah.
Which kind of T cells first of all?
All T cells.
All the T cells, both CD8 positive and CD4. So all the responses related to CD4 and CD8 positive responses are affected. So there is a severe T cell deficiency. What about B cells? B cell numbers, first of all. Is the number of B cells normal or abnormal? Number of B cells. Number is normal. But the function is abnormal. Humoral immune response is abnormal. Because we do not have problem in B cell development. We have problem in B cell activation. We do not have T cell help for B cell activation. And you can see here different patients with DiGeorge system here have underdeveloped gene because those patients have a deletion in their chromosome 22. They have a deletion on their chromosome 22. That this deletion affects indeed just multiple genes are deleted in those patients. That's why manifestations other than immune systems those patients have. For example, underdeveloped gene, different ears. You can see that the patient have different type of ears. Indeed, the development of the ears is involved here. White sets of eyes. The distances between the eyes is larger here, and cleft palate is a very common consequences of DiGeorge syndrome.
It's most common in boys.
Yes, it's most in boys. It's more common, but I think yes. I'm not sure. I'll tell you that. And now I will show. Another disease is linked A-gamma globulinemia. What does this mean? A-gamma globulinemia. What does that mean? A comes from absence. Gamma globulin, what is the gamma globulin? Immunoglobulin, another name of antibody. I told you, antibody, immunoglobulin, and gamma globulin. They are the different names of antibody. So we have absence of antibody in the blood. This anemia is from blood. So A-gamma globulinemia means the patient has problem in the production of antibody. Yeah? The most important problem with this patient is in B cell maturation. This is a common mutation that you do not need to memorize the type of mutations. So we have problem in B cell development and maturation. So when we have problem in B cell development and maturation, so patient won't have enough antibody. So have a look here. The amount of immunoglobulin is very low, plus very few B cells. The B cells number is low because we have problem in B cell development. But we have preserved T cells. It means T cells work normally because T cells do not need B cell for their activation. So T cells function is normal, but B cells function is abnormal. So both serum immunoglobulin, those three manifestations are important for detection or diagnosis of X-linked agammaglobulinemia. Serum immunoglobulin is very low, peripheral B cells are reduced, and normal T cell maturation and function we have in those patients. And again, we have recurrent bacterial infection, extracellular mainly, because of the absence of antibody. Treatment is done by IV immunoglobulin. IV immunoglobulin, what is IV immunoglobulin? Injection of IV immunoglobulin to the intravenously. Or sometimes subcutaneously. So it's a passive immunity. It's an example of passive immunity. And that's it. So there is another defection in the lymphocytes, lymphocyte activation. We have problem here with lymphocyte activation, which is called hyper-IgM syndrome. What does that mean? I talked about it already. Hyper-IgM syndrome. What comes to your mind? IgM. Yes. Why the amount of IgM is increased?
Because of decreasing T cell?
No, IgM do not need T cell. The other classes need T cell. IgM is T independent.
Genetics?
Yes, the genetics. Yes. Because there is no normal class switching. Lady, there is no normal class switching. The other classes of antibody are not produced normally. The reason is that any process related to T cell for class switching is defective. Mainly we have problem in CD40 ligand, and some other cytokines, some other genes that I'm not going to the details. Many genes are involved in class switching that you know one of them, CD40, and it's the most common form. CD40 or CD40 ligand deficiency that leads to improper class switching. When there is no class switching, the only antibody in the serum is IgM. So the patient will have IgM syndrome. Again, it has both X-linked and autosomal form of them. So IgG, IgA, and IgE is reduced. Both of them are reduced, but IgM predominates the immunoglobulin of the serum, and it's very important. Cell-mediated immunity is impaired by when we have CD40 ligand deficiency. CD shell is 40 version of 40. So when a patient has deficiency in CD40 ligand or has deficiency in T cell response. Do you remember here when we have crosstalk between APC and CD40, we have MSC co-stimulator and CD40 ligand we have here again. I think we must have it here. No. it is involved in crosstalk between TLPER1 and macrophages. TLPER1 sends the signal to macrophage in two ways, by CD40 ligand and cytokines. When the CD40 ligand doesn't work normally, so the patient will, the APCs will not receive the help from TLPER1. Okay? So sometimes we have some deficiencies in antibody, some kind of antibody production. Some of them is called selective, it's a very common disease, selective IgA deficiency. It means that the patient cannot produce IgA. What problem is possible for that patient? Mucosal immune responses. Yeah, but because IgA is just a small part of immune response in the mucosal surfaces, we have other components, the patient won't have any significant problem. The main problem with selective IgA deficiency is transfusion. The patient that do not have IgA, lady, please, notes over here. We are talking about IgA deficiency. When a patient doesn't have IgA at all, they have problem in blood transfusion. Why do you think it's so? We have a patient with IgA deficiency, and this patient have problem by blood transfusion. If we transfuse blood to them, it will have problem. It will some problem will arise for him. Why? Because, for example, if we want to transfuse the blood from a normal person to a patient with IgA deficiency, in the blood of normal patient we have IgA. When IgA is transfused to the patient, the patient will produce antibody against IgA because IgA is foreign to him or her. Because it,
Antibody against antibody?
yes, we have two persons, person A and B. This one has IgA deficiency, or it doesn't have any IgA, but this person has IgA. When we transfer IgA from the patient from this person to this one, so the A will produce anti-IgA antibody because this patient haven't had IgA at all. So IgA is foreign for him or her. Okay? As the IgA is foreign for it, it will produce antibody against it, and it causes transfusion-related reaction. But no other significant symptom will have. That's why it is said asymptomatic, the absence of symptoms. Common variable immune deficiency is related with decrease in IgA or Ig and other diseases or other antibodies. So the patient cannot produce the antibody for different reasons. The main reasons is not well understood until now. So the common variable immune deficiency is another examples of having deficiency in the production of antibody from different classes. So we have a defective humoral immune response. So you know that we have these cells. These cells will recognize antigens, and after recognition we have proliferation and differentiation to plasma cells, and plasma cells will produce antibody. Several genes are involved in this process. And deficiency or mutation in all of them can cause CVID or common variable immunodeficiency. So we have diminished B cell activation indeed. Defective or weakened B cell activation. You can see here again granuloma formation. You can see here granuloma or the nodular pattern of jejunal mucosa of the patient again. This is a jejunal the part of a small intestine of the patient with common variable immunodeficiency. I've talked about them. So they are the signs, the important signs that few slides are left, please. They are the important signs, the warning signs of immunodeficiencies. You have to study all of them. I have talked about them in the previous slides. So, treatment principles for congenital immune deficiencies. So first of all is replacement of defective proteins. For example, IVIG is a good example. Or replacement of defective cells, for example, hematopoietic stem cell transplantation. Gene therapy is done for the replacement of the mutated gene with normal gene, or that's enough. Now, secondary immune deficiencies, few slides are left. Or acquired immunodeficiencies. The most important one is HIV infection. But there are other conditions that can cause immunodeficiency, including malnutrition or obesity, anything related to protein calorie balance, or cancer, mainly cancers that involve bone marrow. When a cancer engage bone marrow, so most of the immune cells are produced in bone marrow. They won't have normal white BC production or leukocyte production. Chemotherapy and radiation or irradiation of the patients for cancer treatment cause immunodeficiency. Immunosuppressive therapy given to the for patients with transplantation and again cancerous patients, and loss of spleen. Patients sometimes, for example, as a result of car crash, we have to take the spleen of the patient, and patient with loss of spleen. All of these conditions are examples of secondary or acquired immunodeficiency. So about HIV. HIV is a common cause. I'll give you 2 or 3 questions about HIV as an assignment. Please tell to the students to, in Arabic, put in the if you have channel or group, I do not know, that you have to send me the assignment. You should write your answers on the paper. Yes. Do not send me the answers on WhatsApp. You should write on the paper and your name on the paper and take picture and send the picture to me. And about HIV, I won't explain anything. I will ask you 3 questions about HIV, and about that questions, I'll give you question for your exam. Okay? Clear? That's all. I won't explain anything about HIV. I will give you questions, assignments, and your assignments would be anything that I'm going to talk about. That's all. You are tired, you do not listen, and when you